literally is contracting heart muscle that is bioengineered from an ordinary skin sample. And we've done it from 2-day-old kids to a 100-year-old couple. So we have literally shown that you can do it from virtually all age demographics, which is remarkable. Kids born with congenital heart disease are missing parts of their heart, and some of them are even missing half of their heart. It's the most severe form of congenital heart defects that inspire us to use this technology in its most grand form to literally rebuild hearts so that these patients don't have to have a heart transplant and can live a normal lifespan. Exercise, nutrition, and all of these other things probably do this at some level, but that's really where this science is leading us, is to realize not only is it possible, but what is required to make this feasible to happen in our own bioreactors. And when I say our own bioreactors, I'm referring to our own bodies.
Hey everybody, it's Regan Archibald with Unreasonable Health, and we're so glad you guys are here because today we're going to literally be uncovering the topic of stem cells in a brand new way with the world's leading expert, Tim Nelson. He's an MD, he's a PhD, he's a researcher, but he's also CEO of HeartWorks. And Tim and I run in Strategic Coach. He was, we got connected through Dan Sullivan and Joe Polish and a few other people, Mike Koenigs, I believe. And so, Tim, it's so good to have you on the show. Thanks for taking the time.
Yeah, it's great to be here. Thank you.
And you've been swimming in the world of stem cells for a decade, 2 decades. How long have you been in this field?
Yeah, it's going over 2 decades now. I was training to be a cardiothoracic surgeon and got inspired by the idea of stem cells and regeneration. And the human embryonic stem cells were discovered while I was in medical school, and that kind of ignited my interest in it. And then as I was in my training in cardiology, the induced pluripotent stem cell, which is basically the ability to create a pluripotent embryonic-like stem cell, but from the patient's own body. And that's a paradigm shift, right? It's a massive paradigm shift for how our body regenerates, how we think about regeneration, how we clinically think about it, and ultimately how we apply it to future therapeutics. So it's been just an awesome time to be in the field and not only learning from the field, but hopefully contributing to the field.
And you were at the Mayo Clinic. How much pushback did you get when it came to stem cells? Your interest in it at a time where it was in its infancy, and then there was a lot of political issues around it with George Bush and cutting funding and all that. But what was the tension like during that time?
No, it's fascinating, isn't it, to think back and the idea that human embryonic stem cells present a remarkable ability to regenerate virtually any tissue in your body was captivating at the time, right? It was like, this opens up, changes everything. But you quickly realize that the ethical, moral, legal issues of embryonic stem cells were never going to be resolved by science. Right? There's issues that are unsolvable, resolvable. And then it was the induced pluripotent stem cells from Shinya Yamanaka in 2012 Nobel Prize that just blew that out of the water, right? Now you can do all of the magic of pluripotent cells and never make or destroy an embryo, which is just remarkable, right? But even more important than that, for me, it ignited a field, a study that, if we could do this in the lab, this is probably possible in the body, and it's probably going on in our body daily, and we don't even fully know it yet, right? So these laboratory findings are a paradigm shift, but then you got to ask yourself, if we can do that in the lab, can't the body do that by itself? And then that's where it gets really magical, right? That's where you start realizing that all kinds of stuff in our traditional teaching is probably completely outdated. Well, we know it is. And it challenges our dogma and what we think we know about regeneration. So we're just on the tip of this journey right now. I mean, we've learned a lot in the last 2 decades, but we're going to learn so much more in the next 2 decades, and how this becomes applicable to everyday life of how our body heals. I think is going to be fundamentally game changing.
Yeah. Well, and you're at the front lines there. I mean, you've seen it. I believe you've got the first clinical trial that was actually FDA approved for, at least for congenital heart defects. Is that correct?
Yeah, so we in our lab now, we take a piece of your skin, we convert it into induced pluripotent stem cells, and then we make it into your beating, contracting heart muscle. So it literally is contracting heart muscle that is bioengineered from an ordinary skin sample. And we've done it from 2-day-old kids to a 100-year-old couple. So we have literally shown that you can do it from virtually all age demographics, which is remarkable. And yes, now we have the FDA's approval, which is called an IND, to do that by transplanting those bioengineered cells back into a patient's heart to make it bigger and stronger. And that was the first time the FDA gave permission to use these cells for any application in the heart. And that's been a remarkable journey in the last couple of years, and we're still on it.
Yeah, congratulations. And do you notice a difference in the ability for the cells to differentiate in your 100-year-old clients versus your 2-year-olds? Did it take a few more days, or what did you notice?
Yeah, I knew you'd pick up on that one. So it's a fascinating thing. So it really is. So clearly, when you take a 2-day-old skin sample and a skin sample from a 100-year-old patient, the skin cells grow differently. And so it's probably not too dramatic to realize this, that if you cut yourself or scratch your skin, um, and you compare it to the same scratch or cut that your child or grandchild might have, you heal at a different rate. It's just true, right? It just the cells grow differently, and there's some are better than others, if you will. But younger is better when it comes to wound healing, and that's kind of a general truth. However, even though it might take a little bit longer for those skin cells to grow in the 100-year-old, we were able to reprogram back and send them back in time. And so they're chronologically, they got younger. And what I mean by that in skin cells is the telomeres, right? The telomeres are the junk DNA on the end of your chromosomes. And the longer your telomeres are, the younger you are chronologically. And as we age, your telomeres generally become shorter. Well, when you reprogram your skin cells back into the starting material of induced pluripotent stem cells, the telomeres actually grow. They actually
Wow.
elongate. And so you're literally sending your cells chronologically back in time. And, for a biologist, this almost was too, you almost couldn't believe it the first time these reports were published. But it's true. And there is a way of age reversal definitively in cell culture when you make these iPS cells. And that's has some important biological effects on the cells and the products we make, true. But it also conceptually challenges us to think differently, right? I mean, if you can actually force a cell to do that, what's to say that cells don't normally do that in our body? And what's to
Hmm.
say that we can't create an environment where this is more likely rather than less likely? And that's where the field is really at scientifically now, is to debate some of those concepts of things that we've never even been given permission to really think about before.
Wow. Well, and so many doctors still think that, the concept of aging as a disease is just a hoax. And it's like, yeah, don't waste your money. But I see clients every day, and, our mutual mentor, Dan Sullivan, like, he's getting younger and younger from true age testing. When we look at his methyl groups on his DNA and his telomeres, we use a carotid artery scan, so we look at the thickness of the carotid artery, the intima media, and that gives us some ideas of how fast someone is or isn't aging. And I've seen it. I've seen it in hundred, maybe thousands of patients now. And I could only imagine if you can induce. So I guess it's Yamanaka's. He's the one that, and it's called Yamanaka's clock, who essentially the Hayflick limit. Sorry to get technical for everybody here, but the Hayflick limit, Yamanaka was the first one to show, hey, I think there's a way that we can actually start reprogramming the cells. And as you say, we're going to send you back in time. And so what a discovery. And that was 2012. And so that bypassed that big hurdle that was put in place in the 90s with stem cell therapy. And
Right.
you're right there at the forefront doing the hard work of getting FDA approvals. Where do you see this going? As far as who is in your clinic, your clinical studies right now, and then where do you see it going in the future?
Yeah, there's kind of three phases as how I think about it. There's a current, there's a near term, and then there's a long term. And this concept is, as we've been talking about here, of sending the cells back in time, that we can do dramatically in cell culture and definitively prove it, right? I mean, there's no doubt about the fact of what we see in cell culture. It's been reported by many, many people. That phenomena allows us today to send cells back in time, make stem cells, and make those cells then differentiate into virtually any tissue of your body. We focus on the heart tissue. Why the heart tissue? Well, the heart tissue is obviously central to life, right? I mean, the heart and soul of what we do is you have to have a heartbeat every second of your life to be able to have the blood flow, to have everything working. And so it's essential. It's a definition of life. But kids born with congenital heart disease are missing parts of their heart, and some of them are even missing half of their heart.
Mm.
It's the most severe form of congenital heart defects that inspire us to use this technology in its most grand form to literally rebuild hearts so that these patients don't have to have a heart transplant and can live a normal lifespan. So the foundation of what we do at HeartWorks, we build hearts, is making this technology applied to patients with congenital heart disease. And because they are the most high-risk patients, and because they have no options, the FDA is a wonderful partner with us to be able to show that this technology can be safe and it can be effective for literally rebuilding the heart. And that's the current journey. That's the current reality. This study is open today, and we're recruiting for patients with severe forms of heart failure due to congenital heart. And that's where we have started. Now, the next near term, where does this take us? Is really all patients with pump failure. This is game-changing technology that is at our fingertips right now to be able to rebuild your heart and make your heart stronger. And to do it in the lab and then transplant it into your body is the way we can envision today, making this possible. Just a couple of numbers here to make this real. So you're born with about 1 billion heart muscle cells that beat and contract when you're born. And as you
Yep.
and I sit here in our fourth, fifth, sixth decade of life, we have about 4 billion. So there's a fourfold increase in the number of cells that beat and contract in our heart as we age. They grow dramatically when we're infants. In fact, they double in number in the first year of life. But by the
Mm.
time we're in our third, fourth, fifth decade, our heart stops regenerating at that massive scale. And we have about a 0.1% turnover of heart muscle cells on any given day. And that's the average estimated across the population. So if you have a heart attack and you lose a wad of your heart muscle and your heart function decreases to the point you have pump failure, we don't have a way of dramatically improving the heart function in a short time span because our body hasn't learned yet how to regenerate that massive quantity of cells. Well, 2 weeks ago, we just produced our largest run of cardiac cells from one patient we've ever done. And we made 3.5 billion cardiomyocytes in one run. So we literally made the same number of heart muscle cells in the lab from a patient's skin biopsy that's in their heart muscle as they live today. So you are literally making these cells at a scope and scale. Now, it takes 9 months to do that. So it's not like we can do that overnight. But this near-term phase is being able to apply this technology in the lab in a setting where we can create enough heart muscle to be able to surgically rescue and remuscularize your heart muscle. We envision that that's going to be possible in our lifetime. We envision that that's what this technology is leading us to. But then ultimately, where does this lead us? Longevity is a big deal for us all, and some of
Mhm.
us are thinking about it more and more. And you can't destroy your body, treat it with toxins, be a couch potato, and then magically expect biotechnology to rescue you when you're
Yeah.
when you're in a crisis. That's crazy, right? You also realize that despite all the best efforts that some of us do, our nutrition, our diet, and all of the supplements that we can possibly do, some of us have a genetic problem that causes heart problems, and longevity becomes an issue. So the sweet spot long term for us is, how do we identify the gaps that our body has in order to regenerate tissues such as the heart, and be able to supplement that and actually be able to rebuild the heart so that you never have heart failure or pump failure, no matter how long you live. And so this idea of regeneration of the body outside of the body is going to be very relevant for congenital patients, for patients that have heart attacks, but ultimately heart failure is perhaps one of the main reasons that longevity is hard to imagine, because the heart ultimately fails. Well, if we can regenerate the heart, now we fundamentally change the equation of what longevity could look like.
Yeah, and what I've been sharing with my audience too is if you guys are regenerating the heart, then who's to say we can't regenerate kidneys, cartilage, brains? Is anyone doing anything like this for brains that you know
They,
of?
they are, right? So in fact, there's two protocols that just got released for Parkinson's patients, which Parkinson's is a very specific neuron that produces dopamine in a very specific part of the brain. And when you lose those cells, you get movement disorder known as Parkinson's disease. Well, there now are two trials that are open where we can use the same technology, make skin cells into stem cells, and then rather than heart muscle, convert them into dopamine producing neurons and transplant that back into that part of the brain with the goal of reversing Parkinson's disease. So, from ocular diseases to Parkinson's to heart disease to all of the tissues that you referenced, there's a day where this technology in the lab can regenerate these tissues that can be transplanted back in. But then,
Wow.
when you really want to get crazy and think long term and think, wow, what can we do with this? It's really, how do you now do that in your own bioreactor? In other words, in your own body, and
Yes.
train the body to do these things at a
Yeah.
time scale that's relevant. Now, exercise, nutrition, and all of these other things probably do this at some level, but that's really where this science is leading us, is to realize not only is it possible, but what is required to make this feasible to happen in our own bioreactors. And when I say our own bioreactors, I'm referring to our own bodies.
Yeah. Yeah, because that's the key. I mean, even fasting has been proven to show that your body can get younger and you start getting different stem cell expressions. And we've seen this, I've done I don't know, 40 different umbilical cord allografts, and it's been awesome. I feel younger, my test results show better, my joints feel healthier. But having our own bioreactor, especially if it's scientific based, where it's like, hey, Reagan, maybe how do you perceive it? Would there be like you take a skin graft and then we induce it to, like, maybe we do my liver 1 month and then my kidneys the next month and then my brain the next month? I mean, how does it look? What do you foresee?
I mean, the things that I can foresee today is that, we can do these ex vivo, meaning outside of your body in the lab, and be
Yeah.
able to transplant that back in, right? That's the current state of how we can do this. But as we refine those mechanisms, as we understand that better, I don't know how we would do it, but it's not hard to imagine that understanding those pathways will allow us to target the same fibroblasts. Let's put it this way: the fibroblasts in your heart is about a third of the cells of your heart are fibroblasts. They're the
Yep.
glue that holds the tissues together. Those are the cells that we reprogram in the lab to become new heart muscle cells. So
Wow.
why not think of the cocktail, think of the drugs, think of the gene therapies that we could use to trick or to trigger the cells in your heart that are not heart muscle cells to become heart muscle cells, right? And
Yeah.
there's groups that are working on this. This isn't science fiction. This is work in progress. The issue is, how do we get enough cells at enough time frame for it to be functionally significant? That's where the science of it right now is the real challenge.
And that's where peptides have been a big part of our practice for about 10 years, like BPC 157, increases fibroblastic growth factors, thymosin beta 4. Those have been just phenomenal, even some of the growth hormones, secretagogues. And so, we've been taking the approach that, hey, peptides are a way you can turn on some of these genetic pathways and essentially reprogram your genes to get some stem cell expression. And I mean, it would be so nice if people didn't have to inject themselves every day. But even if they are, I mean, and working, from externally or ex vivo, as you called it, is the more proper term. But using some ex vivo stem cells in the interim is like that could be an awesome way of upgrading. And then it's almost like we become our own 3D printers of stem cells in the future, and I think that's where you're imagining the future lies for longevity.
100%. So, I like to kind of put it in perspective. The easiest surgeon on the easiest job for any surgeon on the planet is an orthopedic surgeon. Why? Because what do? You fall skiing, you break a bone, you go to an orthopedic surgeon. What do? They put pins and cast and put the two broken ends back together, and then tell you to
Yep.
come back in 6 weeks, right? And magically their medicine cured you. Well, the reality is the carpenters, put my hip back together with three leg bolts when I broke my hip skiing and made me not walk on it or put any weight for 12 weeks. And miraculously, the body regenerated and healed itself, right? So
Right.
we call this medicine, but it's really not medicine to put carpenter screws in your body. The medicine is the body, the bioreactor that heals itself. And, there's some tissues that don't regenerate. Right. I mean, there's probably some tissues that won't regenerate ever in our body, but there's most of them will. Here's an interesting statistic. I don't know if this is ever been proven, but it's been extrapolated. If I asked you, how old is your body
Yeah.
on average, how old do you think it would be?
Oh, well, I mean, there's so many different systems, but like
Right.
cardiovascular, I'm testing as a 33-year-old. My telomeres, I'm testing at about a 27-year-old. I mean,
Right.
yeah, I
So,
don't know.
so you and your audience get this idea that your body isn't your body, right? Isn't the age that you were born in. So the story I always like to share with people is those of us born in the late 60s, early 70s, are the greatest generation for regenerative medicine. Why? Because carbon-14 was the highest level in the atmosphere that it's ever been recorded because of above ground nuclear bomb testing. Now, carbon-14 is an inert carbon that gets incorporated into any living system that has carbon in it. And carbon-14 it doesn't cause any harm. It's an isotope that basically allows us to date the dinosaurs, right? You take a carbon sample and you can isotype. Well, because it was the highest level in the late 60s, early 70s, ever since then the carbon-14 levels have decreased every year to this year. It's the lowest it's been in my lifetime. So if you take a cell out of my body and you do a carbon-14 date on it, you can predict with scientific accuracy the age that cell was born or the age that cell incorporated its carbon into its structure. Okay.
Yeah.
So if the cell was there when I was born, it's going to have a super high level. If the cell was born yesterday, it's going to have a super low level. So you can do the experiment, right? I don't want anybody to volunteer for this experiment, but if you ground up your entire body and did a carbon-14 dating of your body, you could get a very accurate estimate of your body. And because there's cells like our red blood cells that turn over every 3 months, we have an entire new blood supply of red blood cells, right? That's why we do a hemoglobin A1C. If you did this, it's about 7 years is what people have estimated that our body, on average, is about 7 years old because of the tissue that's turned over, right? And that's in the average body, right? And that's not somebody that's optimizing it. That's not somebody that's doing all the right things. But it's remarkable, the thing. That's a paradigm shift, right? That our bodies are
Fascinating.
not as old as we generally think of them. And so
Yeah.
you have to ask yourself, what did I do in the last 7 years to cause this problem, right? And if I fix it in the next 7 years, I can get over it. So, this is the paradigm shift. And the case in cardiology that some of us recognize is when we had patients that had chemotherapy to treat cancer. Adriamycin is a classic chemotherapy that we use in lots of lymphomas and leukemia, Hodgkin's lymphoma. When we treat those patients with high doses of this chemotherapy, they inevitably develop heart failure in the next 10 to 20 years. And in many cases, it's heartbreaking that we cure the patient of their cancer, but then 10, 20 years later, they develop this unexpected heart failure. And what the scientific literature now is telling us is that because our heart regenerates at a slow level, when we use these particular drugs that completely wipe out the stem cells of our heart muscle, our heart never regenerates. Our heart becomes older, it doesn't rejuvenate. We develop these heart failure symptoms with time. And there's certain drugs that have a more potent effect on our heart muscle cells than others. And adriamycin is the classic one that we use for a lot of chemotherapies that ultimately ends up causing heart failure. So it's another way that we can see the
Mhm.
medicines that we take might cure one problem today, but might cause a problem down the road because we're essentially wiping out the ability of that organ to rejuvenate itself. So all of these things are starting to teach us more and more about how we can take care of our bodies in a different way. But then if we do have the crisis, how do we ex vivo use these cells to bioengineer replacement parts? And it's going to take both, right? It's going to take all of the above to have the health span that we all desire.
Yeah. Well, and just back to the 7 year testing with carbon 14, I think that's because we're losing trillions of cells just as you and I are speaking here, and it's the stem
Exactly.
cells that are repopulating those. And some areas of the body, like the stomach and the small intestine, those are going to be repopulating really quickly, whereas
Exactly.
maybe your bones are a little slower, but it's still way faster than we think. It's like we're a lot less matter than what we think we are. And
That's
so
exactly right.
if you go back just to put a little context around that, if we're don't do the experiment, but if Tim was able to do the experiment and ground you up and you're 7 years old, why are we aging then? And that's where the Hayflick limit comes in, where the cells start going senescent. But what are the other things that you're seeing that are causing the deterioration? Because I guess if we understand the cause, then there may be a better solution.
Yeah, and specifically in the heart, why is the embryonic heart able to regenerate more than the adult heart? Some of the mechanisms that are coming to light are the early oxidative stress of the heart muscle. The heart muscle is oxidatively metabolically active, and the free radicals and the oxygen is what seems to be the trigger for fetal hearts that are growing really fast. And then in postnatal life, the oxygen and the free radical species seems to be the thing that turned that off. So
Mm,
there's plenty of thoughts of, well, let's minimize that and let's extend and prolong the ability of the heart to regenerate. But then those of us that are older and past that point, how do we actually go back and reverse it? No, I think there's a lot to be learned yet. I don't think we have that nailed yet. But as you do the cell culture work, you can start getting better insights to how these mechanisms are playing out. and extrapolate that from the laboratory studies to what our bodies are doing today. But that's where the science is at, and there's lots of different opportunities to try to improve this. We're obviously very excited about the ability to fundamentally do this ex vivo and to be able to have that type of a regenerative solution available to people that have heart failure and pump failure that don't have time for their body to recover because of either they're born with missing heart parts or because they've lost parts of their heart muscle. Having the ability to come in and rebuild the heart is another great way to salvage that and get ourselves back reset to have the lifespan that we strive for.
Yeah. Maybe you could share a couple of experiences, case studies. I mean, I hate to call them case studies because it's a clinical term for transformations. Or can you share some things that you've seen so far with your work?
Yeah, so I just told you about the bioengineered cells, which is the latest, greatest, newest thing on the block. But we've had multiple other studies where we've used cord blood and bone marrow derived cells from patients' own body to try to heal and regenerate the heart. And we've treated over 166 patients with intraoperative stem cell delivery into the heart muscle. So it's during open heart surgeries that we are then able to directly deliver the cells into the heart muscle itself. In contrast to an IV infusion or a systemic, we've been able to do that during the open heart surgery. In some cases, we have delivered the cells by an IV as well, and so we've explored all of those type of delivery mechanisms to see what has worked. But we had in a couple of cases that are really remarkable, and it's hard to ignore. One young man that had a failing heart, had a leaky valve, and was very symptomatic, so sick that they couldn't do surgery to replace the valve. And he was not a good candidate for a heart transplant because he had what's known as elevated PRA by antibodies, which prevents him from being a good match to a donor tissue. So he was really in a bad spot. He really had no options. Well, we collected his bone marrow, we processed his bone marrow, and we did an infusion of his own cells back into his heart muscle. And over the course of the next 3 to 4 months, his heart got strong enough. It regenerated and healed itself enough that the surgeons took him into the OR and replaced his valve because his heart was healthy enough to live through the surgery. And now, to this day, he's running around playing basketball and doing sports with his heart with a new valve in it. So,
Wow.
there's examples like that do make us believe that, there's some people that respond dramatically well to these therapies, and yet there's other patients we've tried it on that have not had a positive response, that it hasn't done anything to. And it's really a matter of figuring out why is that? Why do some patients respond better than others? And it's like some people can take Tylenol and it works for their headaches, and other people can't, right?
Right.
I mean,
Yeah.
we just don't understand the patient variation. But these are the examples that do make us continue down this journey of how do we make this more available, and more successful to more patients. But then, ultimately, you and your community are also wired to think, okay, what can we do beyond the open heart surgery before we get there? And,
Right.
how do we apply these lessons learned? And I think that's the opportunity, that it's a regimen of exercise, diet, sleep, all the things that we know we all have to do, to the vitamins, to the supplements, to the stem cells, to the bioengineered ex vivo. And it's really a regimen of all these things that hopefully we can stack together to be able to optimize individual performance.
Yeah, we've just seen remarkable things just in using the umbilical cord tissue, adding that with peptides. And then what I discovered too about 10 years ago is I found it was like a 50-50 shot. Some people would get a really good response, other people not so much. And I didn't like that as a scientist. I said, well, let's rule out the variables. And so I studied the small intestine and learned that if people are eating gluten and dairy, sugar, all the pro-inflammatory foods would actually turn off their body's own stem cell signaling. And so we just started putting people on this 30-day diet, and we had them do more time-restricted eating and fasting. And then our 50% results went up to 80% just by making that lifestyle changes, because it all goes hand in hand. Like, you can't expect, no matter how powerful the cells are that you induce, you can't expect it to have an environment that's really toxic for those cells to thrive. I mean, it's not like we're putting Superman in people's bodies. If you don't remove the kryptonite, then it's not going to be as potent as we'd like it to be.
I think of stem cells because there's lots of different types of stem cells, right? And it means a lot of different things to a lot of different people. But I like to think of it as a kind of a farming analogy, that we've got fertilizers and we've got seeds. And
Yes.
some of our stem cells can function as fertilizers that can go in and stimulate the native tissue to grow better and be more resilient, just like our fertilizers on our lawn or our crops. But in other cases, you got to plant the seed, and just putting fertilizer on isn't going to be sufficient. If there's no seed to grow, the fertilizer isn't going to do anything. And so it's really a matter of understanding the mechanism of disease and applying both the fertilizer and seeds. But then also don't forget that if we don't irrigate it or water it, the seed and the fertilizer aren't going to do anything, right? So we got to have the irrigation system, and I like to think of the irrigation system as the nutrition, the hydration, and all the other things that
Blood flow.
Body, right? And so it's really a matter of putting all of these things together. And in the most severe cases, we're not going to be successful if we don't have irrigation, if we don't have fertilizer, and if we don't have the seeds. But in some cases, irrigation and fertilizer might be just sufficient to be able to do it. But I like what you said, is that the pro-inflammatory environment in our body is a counterbalance to the regeneration. And we could talk at length about why we have both, and there's always good and a bad and everything, right? But the reality is the inflammation does cause a challenge for regeneration. And so sometimes our fertilizers are acting primarily as a anti-inflammatory. And just knocking down the inflammation is sufficient to allow the endogenous seed in our body to regenerate and to heal. And so I think thinking of this holistically is really, really important. And hopefully we as a scientific community at all levels are willing to learn from each other and adapt and incorporate, because it takes all of us together to ultimately figure out this complex series of regimens. But I love the idea of getting a better nutritional status for patients that do our stem cell therapy, because if you put the fertilizer on a rocky, dry soil, it's not going to do a damn thing, right?
We have about 25,000 patients to prove it. So this has been so fast. I love your knowledge, Tim. It's just refreshing to hear what you've done and taken the work that we read about, and I've heard speakers talk about it over the years, but to see you put it into action in such a, doing it the right way. I love it. And maybe you could talk a little bit. We have a lot of entrepreneurs on here, people who really want to push longevity medicine forward. I know HeartWorks, you guys you have donors. If somebody would love to donate to your cause, what's the best way for them to do that?
Yeah, so HeartWorks is a nonprofit with a really unique structure. And so we like to think that philanthropy is underutilized to move biomedical innovation forward. And in fact, I believe philanthropy has to be a catalyst to be able to move some of the most high-risk, most exciting technology forward. But if we don't build a sustainable business model out of that, we're ultimately going to fail, because if it's just a charity that requires the hat to be passed on a weekly basis, we're not going to be able to sustain and ultimately change the trajectory of these conditions for patients. So HeartWorks is a nonprofit. We do accept donations, and we do collaborate across multiple academic institutions to do the clinical trials for congenital heart disease. And in addition to that, Regen is a wholly owned subsidiary of HeartWorks. That's a for-profit that allows us to anchor the know-how, the IP, the manufacturing capabilities so that we can also scale this to bring it to everybody with pump failure that's beyond the congenital heart world. So we believe that HeartWorks is a very unique structure that can level philanthropic donations as well as investments, and that we believe that this is a unique platform that allows us to move very quickly through the FDA process, through the regulatory process, and get products approved so that we can use them in our clinics at scale. And so anybody that wants to be part of this journey of curing pump failure and helping us move as rapidly as humanly possible, I'd welcome you to check us out at webuildhearts.org. So we build hearts, all one word, webuildhearts.org. It's pretty self-explanatory what we do.
Yeah, I love that. Well, yeah, and then the thing I'm really excited about too is when it becomes available commercially, and then clinics like mine or my Go Wellness Network, we can start deploying these in our hundreds of clinics throughout the country. And yeah, I mean, you're doing life-saving work, and I think you thinking through all the lenses has been really refreshing. So thank you for the time, and keep up the great work. And we got to get you back on in the next 6 months or something to just keep us caught up here.
I'd be happy to join you and keep you updated. And it's a journey that we're on, and I appreciate you and your audience. And it's going to be a remarkable next decade here in front of us. There's no doubt about it.
Yep, best is yet to come, no doubt. Well, thanks, Dr. Nelson. And for those of you who love the show, make sure you share it with people who you care about. Look at webuildhearts.org. Make a donation. I think it's one of the best places you could put your resources that's going to a great cause. So thanks again, everybody. We'll talk to you next time.